Okay, right. So let's continue with the second LU. Previously we have covered LU1, right? We talk about scientific communication and also so the importance of scientific communication. What is scientific communication? What is communication first? And then what is scientific communication? How science depends on the communication. Okay, so that's what we have discussed previously. So now let's move to the second LU. So in the second LU, this is the ELIP. So we will talk about what are the steps in a modern scientific investigation. That will be covered in module 3, which is today. So next week, we're going to discuss about module 4, which is on thesis research project. Okay. So today we are going to focus on what are the steps in modern scientific investigation. Okay. So I've uploaded the lecture notes on ELIP. You can check out. Um, Hold on. Okay. I hope that the lecture slide is clear to you, even though it's a bit smaller, because I want to sketch something. Okay. Okay, so what is this module is all about? Um, so these are the subtopics. Okay, so talking about steps, it is a sequence of actions. Yeah? So the steps or the procedure in executing a modern scientific investigation. So that is what we're going to discuss. And there are several steps, and altogether there are 7 steps that we are going to go through. Okay. So why it is important to have all the steps? Remember, if you remember the definition of research in general, yeah, not just for the scientific research. Research is a systematic process. A systematic means you have to to execute the things step by step, properly, right, with a good planning and so on. So these are all the steps that have been taken, yeah, in executing a modern scientific investigation. Okay, when we talk about modern, because science is not a new thing. People decades ago or centuries ago, they have practiced science, but perhaps some of them may just do it without proper documentation. So that's why in modern scientific investigation, the scientists define the steps and outline what are the things that need to be taken into consideration. And these steps are all what what I have practiced as a scientist. Yeah, and also what other scientists have practiced around the world. Okay, we start with step 1 and we end with the final step, that is to communicate the science at the end. Okay, all right. So let's have a look at all the 7 steps briefly, and after this we're going to discuss one by one. So the first step is to identify the question and define the problem. Before that, is it clear? My voice and everything? I hope you can see the screen.
Ya,
Yeah, it's a
Dokter.
bit
Yes,
smaller.
Dokter.
Yes, Dokter.
Okay, thanks. Okay. So the first step is to identify the question and define the problem. Okay. So if you remember, so in the second screen, you remember this is what we call hourglass, right? Hourglass. So if you invert the glass, I mean, yeah, I just want to take the shape, the shape how it, which I want to relate with our topic today. Okay, So the problem, see, this is if you see the surface over here, right? It's wide, it's broad, yeah, it's broad. Yeah? So means that when we want to do any research, we start with a broad topic, a broad concept, okay? And yeah, that is where we start to identify the problem, yeah, first. But at that point, the problem is still general, it's still something that is not specific yet, okay? So identify the question, define the problem. So if let's say if you want to relate with your FYP later, final project, because that is the nearest your nearest objective or target, and it's a research. FYP is a research, yeah? Because you are going to do an investigation, right? And you have to plan, and it involves a lot of experiments. So it's a research. Okay, that's the easiest for you to relate at the moment. So whenever you get the topic from your supervisor later, you will have to understand what is the topic is all about. Okay, so when we are doing research, we are actually narrowing it down. Yeah, so biotechnology is a broad field. You study a lot of things. You study about microbiology, biotech. You study about animal biotech, plant biotech, industrial biotech. What else? There are so many. Bioinformatics. But when you do research, you cannot do research in all of the fields because of the limitation of time, energy, and yeah, perhaps you want to do one thing at a time. Maybe you can do lots of things, but it takes time. Okay. So for FYP, you have to focus on one of the niche areas of biotechnology. So from now onwards, you have to get ready with all the niche areas. When I say niche areas, niche is something, the subunits of biotech. As I said just now, biotech is um, I'm just giving an example directly to biotech because all of you are in biotech. So the niche areas, okay, the subunits of biotech, there are animal biotech, okay, animal biotech, plant biotech. Then we go microbiology, where you study about the microbes. Yeah. Plant, you talk about the plant tissue culture. Yeah. And then there is also bioprocess. Yeah. So at least you have to know these subunits. And because later on when the FYP topics come out, so you have to write the topics. You have to compete with your friends in order to get the best topic or the best that fits your interest. So before you choose the topic, you must know the niche areas. You must know the subunits, because like us at biotech program, we have our own specialization. We cannot master everything. Like I'm not an expert in animal biotech. I'm not an expert in plant biotech. I'm specializing in fermentation technology or bioprocess, the field. So so what the topics that I would offer later to my FYP students is based on my field. It's based on my specialization, which is bioprocess or fermentation. And the same thing goes to other lecturers in the biotech, like Dr. Chung is an expert in animal biotech. So he will offer topics related to animal biotech. Okay. So in order for you to, to choose the topics, that is another step. First, you have to know what are the niche areas of biotech offered at UNIMAS. Okay. So I'm telling you now so that you get ready. You know what you investigate by yourself. What is animal biotech? What is plant biotech? Perhaps you have studied some of the courses. You have studied animal biotech, right? You have taken the course, plant biotech, plant tissue culture.
Yes,
Yeah.
and currently.
Yes, okay. So recombinant DNA technology, okay, that is also another branch. If you like cloning, if you like genetic engineering, then you should approach the right person. Okay, and then bioprocess is me. Dr. Dayang Salwani, Prof. Tawal, some of the Dr. Miki as well. So you have to know who is an expert in those fields. Okay, right. The reason why you have to know the niche areas and you have to know the interest is because when we do research, we will spend lots of time for that. Okay, like FYP, it's not like other courses. It's not like any other courses that you take in second year or in the first year, because you have to deal with it most of the time. Yeah? FYP1 is 3 credit hours and FYP2 is 5 credit hours. So means that the weightage of time that you spend will be more actually than other courses. And you have to do the experimental works at the lab. You have to like it. Yeah, you have to like it in order to motivate yourself to proceed with it. Okay. So in order to for that you have to know your interest. Maybe you cannot expect yourself to like every niche areas of biotech. I myself am not a fan of some of the things in biotech. Like I'm not an expert in recombinant DNA technology because I might not really favor. I know it's important, but I might not favor. It's different. Okay. Like you guys now, you have to study all. Okay, but you have to know what is your the biggest interest in what field. Okay, so that is optional. No one has the right to force you to like animal biotech or other course, other other niche. So it's up to you. Maybe you prefer or you like plant biotech more. Maybe you're interested to know more about how to solve the the problems or the environmental problems using plant tissue culture. So that is like personal interest. So in order to develop that interest, you have to understand, you have to know the field. You have to know what is animal biotech all about, what is plant biotech. Okay. So the reason why I'm emphasizing here is because later on when the FYP topics come out, I think by end of this semester, because this is your second year, second semester. So next year you are going to take your FYP. So normally the FYP topics will come out at the end of semester 2, year 2, okay, which is this semester. So you have to get ready for now, okay? Because as I said just now, like the research is something, it takes lots of commitment, okay? So that is the first, I mean, that's the thing that you have to take note from now. Okay, where am I? I think I'm going too far already. Oh, I want to say that, okay, define the problem, identify the question. So whenever you get the FYP topics, okay, let's say your interest is in bioprocess, okay? Right, you get the topics and from the topic given to you, okay, you have to start investigating about the background of that research, particular research, okay? For example, if let's say you take the project about bioethanol fermentation, right? Bioethanol fermentation. So let's say this one, but you take bioprocess and you take like bioethanol. So you are actually going to narrow down your scope. Okay, now from biotech is a wide area, then goes to bioprocess, which is one of the niche areas, right? Okay, so it's going here. And in bioprocess, of course, there are so many topics, and one of them could be bioethanol. Okay, so now you are narrowing down to bioethanol. So you can see that actually the scope of your research is going to be like narrow and narrow, okay? From the surface over here to whatever the things that you are going to focus on, okay? So you have to identify, regardless of any topic, it's not just bioethanol, regardless of any topic, you have to identify the background of the research. Okay, for example, bioethanol, what is the background of bioethanol? It has been produced by many researchers before, but what is actually the background, what they have done, what is not yet being done, okay? So you have to know the subject matters. I mean the background, yeah, the background. So when you study the background, this is done, how you know the background is you have to read a lot of articles. That's how you know what's going on in bioethanol research, okay? You have to read a lot of it. The more you read, the more you know. So from your reading, you will identify, okay, some people have done this, some people have done that. Okay, so that is the background. And what is meant by here? Identify the question. Identify the question and define the problem. Okay, remember that when in our first lecture, when we talk about doing research, we are not we are not entirely repeating other words. Of course, we have to refer to other words as the references, but we are not doing the same thing completely. Okay, so means that if you are doing research, this is the Venn diagram. This is your research. Okay, this is, let's say this is other research. This is not the right one. Okay, this is your research. This is not you are supposed to do. What you have to do is something like in the other diagram, right? When I say that this is your research, okay, this is other works, other works, other works, and this is yours. Okay, so you have to have your own. You must have the novelty in your research. Okay, so the one that is not overlap. For example, here, the one here. Okay, the one that is not overlap, that is the gap between your research and other research. What is mean by gap? The gap is, it means that the difference means that the things that is not being done by other researchers. Okay, for example, in the case of bioethanol, you have, when you study the background of bioethanol research, some people have optimized the media. Some people have perhaps study about different microbes to produce it. Okay, but within that media also is, I mean, there are many things inside the media that you have to know what are going on. Like, for example, media, let's say, people have used different agricultural waste, but you discover that no one reports the use of banana waste as a substrate for producing ethanol, for example, okay, banana waste. Okay, so that is actually the gap. That is actually the thing that is that needs investigation. Yeah, which means that it can be investigated further. So if let's say you figure out that that is the area that is not yet being explored, and you want to focus on that, okay, so that will be your another niche areas, you know, you're going, as I mentioned just now, from biotech to bioprocess to bioethanol, and even within the bioethanol, there are things that you want to focus more, okay? Maybe you want to focus on the use of banana waste as a substrate, as an alternative substrate for producing bioethanol. So that will be your next focus, your next target, okay? So see, remember, this is the broad concept. So you are going from point to point, you are narrowing it down more and more, okay, your research. Okay, so now let's say banana waste. And in banana waste also, maybe some people have done it, but what is actually the things that is not being done? Okay, maybe people have not yet explored the use of banana waste is quite wide. Huh? I mean, the components of the waste. So maybe one of them is the fiber. The fiber is not being yet explored, for example. So you are going narrow, I mean, you are going to narrow it more and more. So that is actually the question or the problem that you want to identify in the first step. Okay, I hope it's clear. And yeah, this is actually, if you go here, this is the this also represents the one that is the gap. That is also represents the problem that we want to solve in the research. Okay, so when we do research, we want to solve, we want to find the answer to the question that is not being answered before, and we want to solve the problem, perhaps. For example, we want to find the solution of, let's say, how to use banana fiber, for bioethanol production. Okay, so that is what is meant by identify the question and define the problem. You have to go to the point where that is actually the things that you want to study. Okay, so how you want to know that? How you want to know the background? By reading, okay, by literature review. That is a process of the reading. So through reading, you know more and more. So if you don't read, perhaps you do not know about what people have done in bioethanol, or in other research. Yeah, so it's through reading. Okay, so I hope that's clear. So that is the first step. Right. I don't go through the points. I just want to emphasize based on examples. So I think I hope that is clear. Okay, it's fine, right? So identifying the research problem. You need to find a question, unresolved controversy, as I mentioned, something that is not being investigated, something that's not being explored. Yeah. A gap in knowledge. So that is a gap. So in research, we always emphasize the word gap because that is the thing that we want to look for. This gap, research gap. A gap is like this one. This is actually the gap. This is the gap, the one that is the area that is not overlap with other research, your research, yours, and this is other research. Remember the diagram in the last lecture. Okay, so if there is no gap, means this one in this case, the first diagram here, like this is your research, and this is other research. There is no gap. I mean, it's just a redundancy. Yeah? So, okay, that means no gap. Okay, the characteristics of a suitable research problem. It needs some time for investigative experimentation. The problem should be significant. Okay, there must be something that is useful come out from your research. Okay, for example, in the case of just now, banana fiber, if you want to investigate the feasibility of banana fiber as a substrate for bioethanol production, what is the significance of it? So banana waste is a waste, right? So if we can convert the waste into valuable product like bioethanol, bioethanol can be used as fuel, yeah, as a fuel for vehicles. And what is the difference between biofuel and the synthetic fuel? Biofuel is much greener, yeah? And it doesn't emit much pollutants like the synthetic fuel. So that is the significance. So that is the and when we use cheap or waste is you can get it easily, right, without a high cost. So it means that you can reduce the cost of the production. So that is the significance of the research. I mean, the one that here, I mean, the bioethanol production using banana waste. Yeah? So that is what is meant by significant. Significant means what is the importance of it, whether it gives like benefits or not. Yeah? Okay. So I'm just giving an example from this one, but I hope that you can relate with other research. It should require an in-depth study. Okay, so of course, when you have identified what are the things that you want to investigate, so it is time. I mean, in order to know the potential of banana fiber as a substrate for bioethanol production, you have to plan your work from there, and it is time for you to do the research. Yeah? Okay, information for the study should be accessible. Answers to the question can be obtained after the study. Yeah, so after you do the research, then you will know what is the potential of banana fiber. Is there any benefit or not, or is there any potential? Right, problems can be stated clearly and concisely. Okay, so basically that is what is meant by identifying the research problem or the research gap, actually. Yeah? So that is the first step that you have to do in doing any research. All right, the second step devise or form a hypothesis. Okay, so remember the word hypothesis. What is hypothesis? You have studied this in statistics. What is hypothesis? Guys?
It is an assumption or a prediction done about the conclusion of a research prior to conducting it.
Yes, correct. Okay. Assumptions about the outcomes of the experiment or research before you do the research. Okay, before you do the research. Why it is important to form a hypothesis? It will give you a picture about the outcomes. Okay, when you do the research, you must have some expectation about the research, whether or not what happened. Okay, and in science or in statistics, we divide the hypothesis into two types of hypotheses. Who remember? What are the two types of hypotheses? The two types of hypothesis.
Null hypothesis and alternative hypothesis.
Correct. Okay, thank you. Null and alternative hypothesis. Alternative or we call also like research hypothesis. Okay. Right. So what is null hypothesis? What is alternative hypothesis? Null hypothesis means there is no effect. There's nothing happen. Even though you do the experiment, there's no change. There's, like means that the result is there is no effect. That is null. Nothing happened. So that is null hypothesis. So it means that if, let's say, in the case of bioethanol just now, if I want to study the use of banana fiber, okay, right. My null hypothesis would say that there is no, how to say that? Maybe you must have the control as well. The control means that the one that you want to compare with your experiment, right? So it means that if, let's say, I want to compare my experiment, one is using synthetic media to produce ethanol, and one is using the banana waste, okay? So of course the control will work as it is because it has been done before, and yeah. But for the banana waste, if I use it, what will be, what the null hypothesis would be, there is nothing happened. There is no production of bioethanol using banana waste. That would be the null hypothesis. Okay, but the alternative hypothesis is the positive outcomes, like the things that will happen. Okay? There is a bioethanol production using the banana waste, right? So that is the difference between the null and alternative hypothesis. Okay, let's have a look at the points over here. Hypothesis is a claim or assumption about a population parameter. Okay, this is statistic definition. Okay, so basically it's an assumption about the outcome of the experiment. Okay, so there are two types of hypothesis, null and alternative. Yeah, I've mentioned about it. Okay, this is more to statistic definition, but if you want to relate just now with an example of bioethanol, that is the thing. So for null hypothesis, there will be no production using banana, there will be no bioethanol production using banana waste. And for the alternative hypothesis, there will be a production. Okay, so it's something that is to state the possible outcomes. So the null hypothesis is also needed because in some cases it might be true. It might be true. Okay. So at the end, you will see that which hypothesis you will accept is based on the experiment. Okay. So sometimes the null hypothesis may be true. Sometimes the alternative hypothesis may be true. So that's why we have these two hypotheses. Okay. Right. So the third step, okay, from the hypothesis, you will predict possible observations or results. Okay, so in the case of bioethanol from banana waste just now, right? Okay, let's say I put hypothesis over here. Let's see here. So the null hypothesis means that there will be no bioethanol production using banana fiber. Okay, see, you can see that the word no production. So alternative hypothesis, there will be bioethanol production using banana fiber. Okay, so when you say that predict possible observations or results, so you can see from the hypothesis, let's say in the case of the alternative hypothesis, there will be bioethanol production using banana fiber. Okay, so the observation that perhaps might arise from here is that, what is the condition that favors low production, or what is the conditions that favor high production. Okay. So you can see that under the alternative hypothesis, even though you say that there will be bioethanol production, is a general statement, but under that you can elaborate more what you expect from it. Okay, of course there will be ethanol production, let's say, but what type of production? Is it low? Is it moderate? Is it high? Okay, so that is the elaboration of the observations that you can predict from your hypothesis. But of course, for the null, there is no production. Yeah, you cannot see anything more. Yeah? But under the alternative hypothesis, you can elaborate more. Okay. So that is what is meant here. From the hypothesis, you can predict possible observations or results. So if you want to say low, how much? How much is low? Normally, bioethanol, we quantify it in the form of gram per liter. Yeah. How much in gram per liter is that low concentration? And for high production, how much? Also, right? In gram per liter. Okay. So that is meant by prediction of the possible observations. Yeah. Okay. All right. So the fourth step, design an experiment that tests the hypothesis by trying to falsify your prediction. So this involves identifying and controlling your variables, determine the procedure of your experiment. Okay, so see, now you have the hypothesis, you have possible observations, right? In the case of this bioethanol production. And from here, you expect that there will be a condition that favor low production of bioethanol, condition that favor high production of bioethanol. So what contributes to that situation is you have to know that you have to set variables. So you have to understand this term, variables. Okay, this is very important. When you do research, you have to know your variables. There must be variables, and you have to identify them. Okay, so for your FYP later, please take note of this. So it will be a very important steps before you start your experiment. You must know your variables, how you control the variables, and also the range of your variables. Okay, now we talk about different terms, so it has its own meaning as well. Okay, variables. Now first variables, then we talk about range and how you control it. How you control the variable. Okay, so but first, what is variable? What do you understand by variable? from the word itself. It means something vary. You vary it. It's a thing that you vary, isn't it? That's why it's called variable. Okay, variable is also called parameter. Okay, is the same thing. Sometimes we say variable, sometimes we say parameter. So when you focus, like here, okay, now you are moving, you are narrowing down. Okay, so when we talk about bioethanol production using banana fiber, and you want to perhaps narrow it down, because when we talk about this, even this topic is still wide. There are things that you can actually focus more and more. Okay, so what is variable? Variable is the things that you want to control during the experiment. Okay, so maybe you want to focus on two factors or two variables, because you just have 4 months for your FYP. The time for you to spend at the lab is just about 4 months, 4 to 5 months. You don't have much time, so you cannot study everything. Okay, so that's why it's very important for you to focus and to set the objective. Okay, that is when you set the objective. When you first you have to set the objectives, and from there you can know what are the variables that you want to control. Okay, let's say you said that you want to study, in this case, you want to study objective 1. You want to study the effect of inoculum density. You know what inoculum? Inoculum is the seed culture. So in fermentation, in order for you to grow or to have the production of bioethanol from the fermentation process, you have to initiate the things by putting inoculum or seed culture. Okay, for example, if you bake bread, right? If you bake bread, roti, so you put the yeast, right? The yeast starter, the starter culture, right? So that is the inoculum. So imagine that if you want to ferment bread, actually you are doing fermentation. Bread is an example of fermented product, and the process is fermentation before you get the bread. So it will not happen. The fermentation will not happen if you don't add the inoculum, which is the yeast, the Becker's yeast. Becker's yeast is Saccharomyces cerevisiae actually in the scientific name, but the common name is Becker's yeast. Yeah, or we just refer yeast because you know, like maybe some people may not know their scientific names. We just say yeast in the kitchen, but that is actually the microbes, and the amount that you supply in the beginning of your fermentation, before you start the fermentation, that is what we call inoculum, the starter culture or seed culture. So one of the factors for fermentation is the inoculum density, the inoculum concentration or density. What it means by this factor is the how much you add the inoculum. Maybe you can quantify it in terms of 10%, 20%, okay, or maybe 15%. 15% or 20%. Okay, normally it's 10%, but maybe you want to study what is the effect of varying the inoculum density, inoculum concentration. So what is meant by 10% here? So let's say you have your culture, the total volume is 100 ml. Let's say 100 ml. So what is meant by 10%? Total volume is the total volume of the ration of the fermentation broth, for example. So if you want to have 10% of inoculum, so it means that you have to prepare 90 milliliter of medium, and 10% or 10 milliliter, in this case is 10 milliliter because the total volume is 100 ml. 10 milliliter would be the inoculum. Okay. So 10 divided by 100 is 10%. That's how you get the concentration of inoculum. Okay. Right. So yeah, so if you want to study what is the effect of varying the inoculum density, whether if let's say you put 10 milliliter in this case, 10%, and then if you increase 15% or if you increase it to 20%, will there be any benefit in terms of the bioethanol production? Okay, so that is an example of how you control the variable. Okay, the variable in this case is the inoculum concentration or inoculum density. That is the variable. You understand? So this is variable. So you can see how your variable, the selection of your variable, depends on the objective that you want to set. Okay, so in research, we cannot study everything because we don't have much time. If you want to study everything, it will take the whole life to study. Maybe at this time you study this, at this time you study this. But if you are given a certain period of time, you cannot study everything. Okay, so that's the fact in research. Okay, so that's how here we have to focus more and more. And even though you study about bioethanol production using banana fiber, but now you focus on one variable. Okay, let's say inoculum density or concentration. Okay, let's say you set another objective. Means maybe you think that within that 4 to 5 months you can study 2 parameters. Okay, so the second objective, you want to study perhaps the effect of temperature, the effect of the incubation temperature. So when we do fermentation, we have to incubate the culture at certain temperature, right? And sometimes some of the microbes, they might just have range of temperature that they like. Okay, maybe from your reading, let's say you are using, okay, let's say you are using the microbes over here. Let's say you are using a new strain. Okay, let's say a recombinant, using recombinant E. coli that can produce ethanol. Okay, right. So that is the background of your research. And you know that the recombinant, this strain, this recombinant E. coli, the temperature that it favors is between 30 to 37 degrees Celsius. That's from your reading. Okay, when you study about the background of research, you get to know that the range of the temperature is between 30 to 37 for this particular microbe, for this particular strain. Different microbes have different temperature. You cannot set one temperature for all microbes. Yeah? So that's why you have to study it specifically. So let's say the idea is the range is from 30 to 37. Okay, that is from your reading, right? So in this work, you want to study specifically. So maybe you vary by 2 degrees, okay? Maybe 30 degrees Celsius, 32, 34 degrees Celsius, 36, or maybe you add another 138, right? So now temperature is your second variable. Okay, this is variable 2. Variable 2. And remember just now I mentioned about range. Okay, for the first variable, the inoculum density, this is maybe the range that you want to study, yeah? From 10 to 20%. Okay? 10 to 20%. So the range is 10 to 20%. The range is the range. This is from the lower value to the upper value of the variable that you want to investigate. Okay, that is range. So for the temperature in this case, okay, this is from your reading, but in reality, you want to do the experiment between 30 to 38. So your range will be 30 degrees to 38 for this work. Okay, so that is the range for the second variable. So that is what is meant by identifying and controlling your variables. First, you have to identify your variables. So in this case, is inoculum concentration and density, and then temperature. Okay, you identify the range, right? And from the range, you know that you will control the variable at that particular values. Okay. Right. So another step in this, another thing that in this fourth step is to determine the procedure of your experiment. Okay. This is actually to be done before you enter the lab, huh? And you will do it in FY31, whereby you find out the information related to all of this stuff. Okay, so make sure you take note. Procedure. What is meant by procedure of your experiment? So means that now, let's say in this case, you know that you want to study bioethanol production using banana fiber, and the microbes that you're going to use is a recombinant E. coli. Recombinant is, I hope you know, is a genetically modified. So microorganisms are divided into wild type and recombinant. Okay, this is, you should know about this. This is the basic things about microbes, genetically modified and wild type. Wild type means the one that is wild, that is natural, the one that we got it naturally from the environment. Genetically modified or recombinant are those that you have altered the DNA, and perhaps that strain carries certain gene that can produce certain protein of interest. Okay, for example, E. coli. E. coli is not really a natural strand that produces bioethanol, but when it is genetically modified, means that the gene that can produce bioethanol is inserted into the E. coli, right? So that it can produce ethanol when it metabolizes certain food. Okay, so that's how it can do the work. So that is just some background about recombinant things. Okay, now the variables that we have chosen is inoculum concentration, and the range is from 10 to 20%, for example. The temperature is the second variable. The range is from 30 to 38. This is your plan. I'm talking about, this is when you do a research, you have to plan. You have to plan the range, you have to plan the variable. And from here, you can expect what kind of things that you want to do. Okay, so that will determine the procedure. Okay, procedure means how you do the work, how you do the, in this case, what you have to do is, what you have to find out is procedure of bioethanol fermentation. But before that, you also have to know the methods, the procedure, how to grow E. coli. Okay, how to prepare the seed culture, how to prepare the inoculum. Okay, so you have to identify like every step of it. It has its own procedure. Okay, for example, in this case, how I can say the flow of the work. We have to know the flow of the work. First, it starts with preparation of inoculum. Okay, and also you have to know that you are using banana fiber. Banana fiber is you get it from the banana plantation. Perhaps you also have to know how you want to prepare your banana fiber. Preparation of banana fiber. Okay, and then you will start with the fermentation, bioethanol fermentation, right? So when you do the bioethanol fermentation, you have to monitor the growth, like the cell growth. You have also to monitor the substrate consumption. And also you have to monitor. The most important thing is the bioethanol production. Okay. So this is the thing that you have to prepare the flow of your work. Okay. So that you know that, for example, for the preparation of inoculum, there are certain procedure for it that you have to find out how, because you know you can see that each of that has its own procedure. Like this one, for the preparation of the banana fiber, what you have to do, perhaps you treat it physically, you prepare it, perhaps you boil it, I don't know, maybe you grind it. Yeah? So each of this, you can see each of this has its own procedure. So that's why you have to know the flow of your work and also to identify what are the things that you need to find out for each of the steps. Okay. And for example, this is analysis. This is under analysis. All of these are under analysis. So how you want to monitor the growth? It's by, usually is we check, we measure the OD, the optical density of the culture growth, whereby during the fermentation, you take the samples every, for E. coli, maybe every 2 hours or every 3 hours, okay? And then you check the OD. Okay, so here you can see that for the cell growth, it has its own procedure as well. How to monitor is by perhaps absorbance measurement. Okay. That is the method. That is the method. Okay, for substrate consumption, maybe you check by using maybe HPLC. HPLC is a, or maybe an easy way like this one is, let's say, DNS assay. DNS assay, whereby this DNS assay can quantify the concentration of reducing sugar, for example, like glucose. Okay, so you have to know what is DNS assay, how it works. You have to find out the procedure for it. Okay, and for bioethanol production, maybe using HPLC. So what is HPLC? So that is another procedure. So you can see that the whole work consists of steps, whereby each of the steps has its own procedure. Okay, so that is what is meant here. You have to determine the procedure. You have to know before you enter the lab, how you're going to measure the absorbance. And from here, you also have to know, you have to identify also what are the chemicals that you are going to use, equipment. Okay, so from here, you can also know the chemicals and the equipment. So of course, when you measure the absorbance, you will be use cuvette, Eden cuvette, the one that you put the culture growth into it, and you put it into the spectrophotometer. So the equipment is a spectrophotometer. Yeah. So for the DNS assay, DNS, it has its own reagent. Yeah. So you have to know what are the chemicals needed in order to prepare DNS reagent, and how you're going to do the assay. Yeah. And this one is also by measuring the absorbance and what equipment you are going to use. So it's actually from the plan of your procedure, you will know the chemicals, the equipments, and yeah, the flow of your work. Okay. So that is involved in what we call the design of the experiment, or the fourth step. Yeah? Fourth step. Any questions so far? This is what you are going to prepare for your FYP, yeah, which is going to be very soon. Right, the fifth step, right, before that, when you design an experiment, right, apart from knowing the chemicals, equipments, and also you have to identify what are the risks. So why is it important to identify the risk before you enter the lab or before you start the work? Because lab is not, excuse me. Sorry. because in the lab, if you don't plan or if you don't identify your work properly, there will be risks, right? There are going to be risks, potential risks in the lab. Sorry, I have like so. Okay, so apart from identifying the chemicals, equipments, you also have to identify the risks. And ideally, researchers need to fill up what we call risk assessment form. So what is this risk assessment form? Is where you identify what are the risks associated with the chemicals used, what are the risks associated with the procedure that you are going to use, whether it's very risky, moderate, or low risk. Okay. And also it's related to how you dispose the chemical waste, for example, or the biological waste. So you have to know beforehand, because later if you enter the lab, if you don't know the risk, or some of the chemicals are hazardous, you might just touch it, but actually it can't be touched with your hands. So what happened? It will be hazardous to you, right? It will affect your health, right? So we don't want researchers to be so passionate, but they don't care about the risk. So risk is a very important thing to be taken into consideration, right? So when I was in the UK, when I was doing research in the UK, one of the important things emphasized at the laboratories is a risk assessment form. We are not allowed to do work before we fill in the risk assessment form, and we have to submit the form to the safety officer. So the reason is because anything happened, like if let's say explosion happened at the lab, you know, you will be questioned why you do not know about, why you do not know about the risk. It might be the accident happened, it happened because of your mistakes. It happens because of you handle the things carelessly, right? So at the end, the researcher will be blamed. And it's not just researchers, the university, the whole university will be blamed, right? Where is the safety officer? Why the faculty is not concerned about that? Okay, you can see the impact is very huge. And I remember during a briefing, during the safety briefing, before I entered the lab in the UK, they say that why is very important. What is the impact to the university? So whenever accidents happen at the lab, okay, it will give an implication to the image of the university, in a sense that that research is actually there are many funders. Funders are those that give money to do the research. So if let's say accident happens because of the research, because of the things in the lab, funders, those funders like maybe the Ministry of Education, Ministry of Science, they will not give the fund to the universities that have the records of accidents at the lab. Okay, that is what being told in the UK. So to that to that serious level that they are very concerned about the safety. So that's why, in order to avoid that from being happen, they enforce the rule. Like they are very strict about safety, about the safety of the researcher, the safety of the work being done, because it will give a very huge implication to the university. So if let's say the funders, they don't give the money to the university to do research, so means that the researchers, the lecturers, we can't do any work, we can't proceed with our research. And what happened? At the end, the performance of the university will be affected. Okay, so that's why the risk assessment is very important. The risk assessment is you assess the risk. Okay, let's say now in the case of bioethanol fermentation, what is the risk of the experiment? Let's say when you prepare the inoculum, is there any risk? Yeah, of course there will be risk, but the difference is whether the risk is low or high. Okay, for example, some of the chemicals in the lab, they are carcinogenic. Carcinogenic means it can cause cancer. So assessing the risk before you use the chemicals will enable you to take precautions. Now, let's say you know that what are the chemicals to be used in this work, for example, this bioethanol fermentation. Okay, let's say one of the chemical is hazardous, is very hazardous, is carcinogenic. So you take care of it, right? Of course, you will not deal with the chemical carelessly because you know that anything happen, it will give bad effect to yourself, right? For example, IPTG. IPTG is carcinogenic. IPTG is normally used for inducing recombinant bacteria to produce the protein of interest. Yeah? I think you might come across this chemical in recombinant DNA technology. I'm sure that you got to know these chemicals. I forgot the full name, but the abbreviation is IPTG. So IPTG is carcinogenic. If let's say you are dealing with this in the future, please take note that it is carcinogenic. So means that you cannot touch with your hands. You have to make sure that you have glove before you touch it, and you have to deal with it carefully. And when you measure the weight, for example, you might have to make sure that there is no spill of it, because if let's say there is spill and the next researcher that used perhaps the balance do not know that that is IPTG and might, you might touch it. So it will actually endanger others. It's not just yourself. Okay. So that's the importance of assessing the risk before you do the work. Okay. And not just chemicals, some of the equipments can be also hazardous. Okay? For example, those that involves pressure. For example, autoclave. Autoclave, do you know autoclave? So you will need autoclave because you need to sterilize your consumables, your glassware. Okay, what is the big no when you use autoclave? When you use autoclave, because autoclave is it is conducted at high pressure, right? High pressure and high temperature. So means that when you insert the things in the autoclave, right? When you, let's say you want to autoclave your media, right? Let's say your media in a bottle. So this is your media. You know the Scott bottle, the one with blue cap. Yeah. So normally we just type it, but when you put the things in the autoclave, right? So if you know the risk of it, you know that you cannot tight the cap very tightly when you insert any bottle in the autoclave. That's why it's very important to know that because you cannot tight it, you cannot close it tightly, the caps. So you have to lose it. So that there is a space, because once the things is autoclave means that the media, the whole thing here will be heated. It will be heated. So there will be a pressure inside. Okay, so if let's say you tight it very tightly, you close the cap very tightly, means that there will be a pressure inside. There is the things will be boiled inside, right? So there will be pressure. So if there is no space for the pressure to release from the bottles, what happened? What happened? Do you think what happened? So let's say, yeah, it will explode. It will break the bottles. So that's one of the risks in the lab if you do not know the rules of using autoclave. Okay, so that's why it's very important for you to identify the risk, assess the risk. So please remember the next time when you autoclave, any bottles, it must be loose. It still can be capped, but you have to lose it. Okay? And also the volume of the media, the liquid inside the bottle, it cannot be full. Let's say this is the limit. You cannot fill it to the fullest. Otherwise, it will be spill out. So there must be a space. Like this one is the liquid. Okay, this is the liquid. What I mean by space is this one. This is the space, right? So because you have to allow some space for the for the liquid when it is heated in the autoclave. Okay, so this is the basic rule. So apart from the chemicals, you also have to know the risk of the equipments that you are going to use in the lab. So that's why risk assessment is very important before you enter the lab or before you start the work. Right. Okay. Yep. Right. So I mentioned about the procedure now. Let me just take some of the points in the slide. Risk assessment, yes. Okay. Selection of the procedure. Okay. I've talked about the accessibility, expertise, safety. Yeah, safety, whether it's hazardous or not. Okay. So in terms of the methods, so how you want to know the methods, I forgot to mention, how you want to know the methods used for the absorbance measurement. I mean, absorbance measurement is quite common. Some of the methods may be like DNS assay or maybe Bradford assay. Okay, so this method is actually proposed by researchers. Bradford is by Bradford. The name of the scientist is Bradford, so that's why he named the method as Bradford assay. So you have to also give credit to the person who invented the method. So Bradford normally we cite Bradford, the original researcher. I think the method was invented in 1956. Yeah, there is a paper about it. If you Google, maybe you can find it. So apart from knowing the procedure and putting those procedure in your proposal, these are the things that you need to write in your proposal. You also have to give credit. This is what is meant here. Credit should be given to who invented the experimental procedure. Yeah. So any method that you plan to use in your work, you have to cite. What is meant by cite is, let's say, in your proposal, let's say you write that the protein will be quantified based on Bradford assay. Let's say. So you have to cite it at the end by putting the reference for 1956. Okay. So it means that here, this statement, if you see, protein will be considered based on Bradford assay. So we know that the readers will know that the Bradford assay is a method by Bradford, which was written in 1956. Okay, so this practice is called citation. You know this already, right? This is what we call citation or in-text citation. Okay, what happened if you don't put Bradford 1956? Let's say you just put like up to here. Protein will be quantified based on Bradford assay. It is as if that the method comes from you, which is not true, and it violates the right of the author here, like the right of the researcher who invented the method. Okay, so that's why you need to cite. This is what we call cite. Huh? Please cite them in your proposal or report. Okay, don't steal someone's standard. Okay. Okay, some of them, this one is not generic. Some of the methods are quite, this is not generic. I will correct it. So some of the methods are quite generic. What is meant by generic method is something to do with like, okay, let's say you autoclave the media. Okay, you autoclave the media. Okay, let's say you put the media will be autoclave before the fermentation, let's say. So you know that what is the purpose of autoclaving in the lab is because we want things to be sterilized, free from microbes. Like for example, in this case, the media. We want the media to be sterilized. So this kind of thing is a general knowledge. Okay, you don't have to cite it. So this kind of statement does not need citation. Okay, this one is meant by generic. This is generic method or generic procedure. Okay, so you don't have no citation. Okay, so I hope that's clear. Okay, so that's all about the research design and variable. Okay, so basically you have to, you have to identify your variable, you have to know the procedure, and also you have to know the risk of your procedure. Okay, let's move to the fifth step. Carry out the experiment to obtain your data. Okay, I'm just focusing on your proposal. That's why in this case I use future tense. You can see here. We'll talk about it later, but just to inform you. Right. So this is what you do. The steps from 1 to 4. This is what you have to identify in your research proposal or state in your research proposal. Or in your case, will be FYP1. Okay. Right. So when you have the proposal, what is the importance of proposal is that remember that research is a systematic process. So we do not want the researchers to start the work without proper plan. Yeah. So with the proposal, it means that you know the background of the research, you know what you are looking for, you know the objective, you know the variables that you are going to control, you know the range of variable, the methods that you want to use, and of course from the methods, it means that you know what are the chemicals, equipments, risks. Means that it covers prior knowledge about the work that you're going to do before you enter it. So those information will be written in the research proposal. And this research proposal, the importance of it, if it's not just FYP, in other situations like us, independent researcher, we also have to prepare research proposal whenever we want to do or we want to propose a research before we start any research. We will write a research proposal. So it covers all the information as I mentioned just now, right? And in your case, you have to present your research proposal in front of your examiners, right? And why you need to present it? Because the examiners need to evaluate whether or not your plan is correct. And if let's say the examiners have suggestions, they can suggest to you, right? And that could be that could help you to improve the plan of your work. Okay, so that is the purpose of the research proposal. So it will be written and it will be presented. Okay, so you have to write and you have to present your research proposal. So once you have the research proposal, so let's say it has been approved, right, by the examiner. Okay, your plan is good, everything, is makes sense. Then that is where you carry out your experiment. You start doing works in the lab. Okay, so you can see that without research proposal, you cannot enter the lab, literally. So the fifth step is to carry out the experiment to obtain your data. Well, you have the plans already, right? Okay, bear in mind, proposal doesn't need to be perfect, but not to say that you don't have to care. But some of the things in the proposal might change. But I can say you just have to try your best to prepare the research proposal. But if there are any changes throughout the project, you don't have to feel bad, or you don't have to you don't have to be so rigid in a way that you have to stick with your plan. Okay, some of the plans or some of the procedure might have to change because of many factors. Let's say maybe you cannot use DNS assay to analyze the reducing sugar, perhaps because of maybe there are some mutations, the chemicals are not there, or maybe there are so many factors in the lab. Maybe you can switch to other method. Okay, so it doesn't have to 100% follow your proposal. Along the way of your research, you can change a little bit. It depends on the needs And also, it should also make sense. You cannot just change it without reason. Okay. Right, so let's focus on the experimental research. So when we do the research, so you have to know that your work is what kind of research, what kind of experiment you are doing, because the experiments in the lab, there are many forms of experiments. It can be, some of them might be just computational research. Depends on the data, and you use software to maybe, to model. Yeah, so it depends. Basically, it depends on the computer. Or some of the research needs needs you to control the variables. Some of the experiments needs you to perhaps compare, and yeah, there are many ways actually. Right, let's have a look at what are different types of experimental research, right? Okay, the first one is the controlled scientific experiment. So what is meant by controlled scientific experiment? The researcher conducts empirical tests while identifying and controlling as many factors as possible that may affect the outcomes of the study. Okay, the easiest way is just now when you have the variable, you control the variable, like what are the variables just now? The inoculum concentration and the density. So you control it from, let's say you conduct experiment at 10%, you conduct experiment at 15%, you conduct experiments at 20%, and you compare the performance of the bioethanol fermentation. So the way how you vary the variable is actually you are controlling it. Okay, so that is an example of controlled scientific experiment, right? Factors is the variable, the parameter or the variable, right? Okay. The second type is correlational studies. What is meant by correlational studies? So just now the controlled scientific experiment, you control one thing at a time. Maybe you want to study inoculum concentration density, right? And see what happened to the bioethanol fermentation. Correlational studies, the difference is that when you study two factors like inoculum concentration density and temperature, and you also see how the inoculum concentration change. What is the effect of the variable 1 on the variable 2? Okay, let's say it means that if you vary both variables, yeah, let's say variable 1 and variable 2, what happened to the bioethanol fermentation, and does it correlate to each other? Does variable 1 depends on variable 2 or not? Okay, so that is what is meant by correlation studies. You want to find the relationship, the correlation, yeah, the correlation between the variables that you study, right? So normally the results is presented in the form of this kind of graphs. You can see the graphs, there are different correlations, right? So let's say here, this is positive correlation, means let's say here, what is meant by positive correlation? When you increase the inoculum, let's say this one is inoculum and this one is temperature. So it means that when you increase the inoculum density, no, it's not. Let me just define, let me rephrase my words. Okay, let's say this is inoculum. Inoculum density, and this is bioethanol production. So when you so what is represented by the positive correlation is you increase the inoculum density and you will get high bioethanol production. So it's positive correlation, is directly proportional. Okay, so that is positive correlation. So negative correlation is when you increase, let's say increase the increase the inoculum density, the ethanol decreasing, the ethanol concentration is decreasing. So that is negative correlation. No correlation means when you observe all your experiments, like all your experiments, there is no specific trend. It's not increasing, it's not decreasing. So like, let's say the data is scattered like this. So we can't actually say anything about the trend. So this is what we call, there is no correlation between bioethanol and, for example, inoculum density. Okay, so that is things that you want to find out the relationship, actually, what is correlation studies is all about. Okay, the third type of experimental research is data collection and analysis. Okay? Some research depends on a survey. Yeah? Survey. This is quite popular in social science. They depends on the data, the response given by the respondents. Let's say they want to study certain behavior or certain trends, and they need the feedback from people, maybe the students or people aged from 20 to 40, like that. So yeah, so it depends on the survey. Yeah? So that is data collection. I mean, yeah, survey. And what they do with the results, they analyze it statistically, and they kind of like derive, maybe they present it in the form of graphs or modeling or something, what they expect, maybe something like that. So it's something to do with the survey. It's not really popular in science. Okay, and then the last type of research is computer modeling. Okay, this one is something to do with the computational works. Okay, some of the scientific works, they are not based on the works in the lab solely. Okay? So maybe they have some works in the lab, but they use the data from the lab that they, from the works that they do in the lab. They use that data to model or to predict. This is what we call modeling. Modeling is when you use whatever the data and you predict what happened to it. Maybe if, let's say, let's say you, for example, inoculum, let's say I don't have any specific example, but basically it's something to do with, like, you use whatever the data in order to predict what happened if, let's say, you increase the range or something like that. So it's something to do with the prediction or simulation, we call it. So some of the research, the nature is modeling, and the nature is modeling. So this one is, you have to use certain kind of software to do the simulation. So this is example of computer modeling result. Okay? So of course, there are interpretation of whatever the software is. Like, for example, in this case, the color here, it represents certain meanings. Yeah. So only people who are in those kind of fields, they understand what it means. So for example, here the red means what, the yellow means what, maybe the density is more, I don't know. Yeah, maybe something to do like with the with the meaning of the data. Okay. All right. Yeah, I'd like to share with you this thought. Creativity is inventing, experimenting, growing, taking risks, breaking rules, making mistakes, and having fun. I like the word experimenting there. So I don't know about breaking rules. I don't blame you for that. But creativity is, experimenting is about creativity. So when you do research, you do experiments in the lab, it also actually develop your creativity. Maybe, like, your plan is like that, but when you start doing works in the lab, you might think something more, and it depends on your creativity. Creativity is something that you think beyond perhaps the scope, the box, right? So yeah, creativity can be also developed while you're experimenting in the lab. So I like that. Okay, so the sixth step is to collect and analyze your data to gather evidence. Okay, now when you do the experiments, so like you do the bioethanol fermentation, it's not just about doing the fermentation without analyzing things. You have to collect and analyze your data. So that is where this step is actually, is analyzes. So bioethanol fermentation is a process, right? And it is that it gives no meaning if you don't analyze it. So what is meant by analyze? You have to know, in this case, you have to analyze the growth, because you are growing the microbes. Fermentation is about growing the microbes, right? To produce bioethanol in this case. So you have to analyze the growth. This is the basic things that you need to analyze during any fermentation. The substrate consumption, right? And also the bioethanol production. So if you don't analyze these three main things, you don't have any picture about what happened in your fermentation. Why it's important to analyze the cell growth is because without the cells, without the growth of the microbes, there will be no products. Yeah. So you must make sure that the growth is there. The cells are growing. So how you know whether the cells are growing or not? You take the samples every certain period of time, like for maybe for E. coli every 2 to 4 hours, right? So you collect the samples, you analyze the absorbance. You collect it and until it finish, until the fermentation finish. So how to know the fermentation finish or not? For E. coli, normally the life cycle is about 24 hours, right? So by 24 hours, most of the cells may be dead already. So the total duration of the fermentation for E. coli is normally 1 to 2 days, 1 to 2 days, 24 hours. So during that 24 hours, you are expected to, as I mentioned just now, you have to analyze the growth. So you expect to see the growth something like this. You study about microbiology, right? What is this? What is this? This is a typical growth profile for any microorganism, for any organism actually. Yeah, typical growth profile, meaning to say that from 0 hour, it will have some time for the lag phase. Yeah? The cells are adapting to the environment, to the substrate, okay? And after some time, it starts to accelerate, right? That is where it's the log phase. And it starts to slow down and finally decline, okay? So this will be reflected in the absorbance, the OD values. So absorbance or OD. So whenever you take the samples, right, you check the OD, an ideal growth, it will show the increase of the OD, the absorbance. So meaning to say that from 0 hour or maybe at 2 hour, you will see the reading of the OD is 0.2, maybe at 6 or 8 hour is increasing to 0.5, and here is 0.8. So they must be increasing. If it is not increasing, it shows that there is no growth. So it means that if there is no growth, in this case, there will be no bioethanol, right? So that's how you know whether your fermentation works or whether your experiment works or not. You have to analyze during the experiment. So that is meant by collect and analyze your data in order to gather evidence. If not, you don't have anything to conclude at the end. Okay? So in this case is cell growth that you have to analyze. And also you have to analyze the substrate consumption. What is meant by substrate consumption? Is the food that you feed to the microorganism, right? So let's say we know that the banana fiber has glucose, right? Glucose. Now we're talking about banana fiber. We know that it has glucose. So you analyze the change of glucose, and how to know that, you also have to take the samples. Normally you take the samples, you keep the samples for all the analysis, for the cell growth, for the substrate consumption, and for the bioethanol production by using different methods that you identified. Okay, for the glucose consumption, why you also have to, in this case, why you also have to observe or analyze, because if let's say it must reduce with time, because the cells are eating the glucose. It metabolizes the glucose in order to produce ethanol. So if let's say, what happened if let's say you don't see any change, right? Maybe you don't see any change of the glucose. It's from 0 hour to 24 hour is the same. So what happened? Perhaps they will not actually get the growth like this. There will be no growth as well. So that's why you have to verify the growth with the substrate consumption. if there is a growth, you should expect the substrate to be consumed during the fermentation, which means that the reduction of the profile. Okay, this is the ideal one. Okay. Another one that you have to analyze in this research is bioethanol production. Right? What is the trend of bioethanol production? You expect it to be increasing during the fermentation. Okay, from 0 to 4 hours, maybe it's increasing something like this. So that will be tally with the growth and with the substrate consumption. Means that the cells are growing, the substrates are used by the cells, and there is a production, because this is the one that you are going to harvest. You are interested with the bioethanol at the end, right? So this is example of analysis for this project. So any other project might have their own analysis. Okay? It depends on the project. So I'm just giving a specific example here. Right. Before that is about the analysis. What kind of things that you have to analyze or collect throughout your experiment. And you also have to know your data. This is all data. This is actually data. The information about the graphs, that is data. You have to know your data, or your work will generate what kind of data. So we study in statistics, there are two types of data, qualitative and quantitative data. So how to know whether your data is which one? It depends on, like the data, whether it's numeric or not, whether you need to describe the characteristics. Right? In this case, it's something to do with the quantity, with the number. So obviously, in this case, your data is quantitative, right? Quantitative. So that's why you have the numbers, right? But you have to know that there are two types of data, qualitative and quantitative. So some works, some research, they will generate qualitative data. For example, if you are studying about morphology of microorganisms of different bacteria, you want to observe it under the microscope. Let's say your research is about screening of bacteria from wastewater samples. Right? So maybe that there are many types of bacteria and you have to observe it under the microscope, right? The morphology. So there will be different morphologies. Morphologies are characteristics of different physical features. So you have to use the right words, terms to describe it. You study this in microbiology, right? So some bacteria, when you view it under the microscope, they are in rod shape. Some are in spear shape, some in bacillus shape. So that is how you describe it. And your data are qualitative because you are describing the features, the characteristics. There is nothing to do with number, right? So that's example of qualitative data. So when you do research, you have to also identify or find out whether your data falls under qualitative or quantitative, because that will determine your final outcome or result or conclusion. For example, in this case, in this bioethanol fermentation, it's not, your data is not, basically it's not qualitative. So if you don't take note of this number, maybe you just observe it and you just record the culture color, which is not correct. So at the end, although you have done your research, your experiments, but the way how you collect the data is not correct and your interpretation about your data is not correct, you might not get the conclusion to your objectives. Okay, so that's the importance of identifying the types of data. I don't have to go through the words by words under these slides, but that's basically what it's all about, qualitative, quantitative. And then the types of statistics. Okay, so there are two types of statistics, or two types of, not just the data, this statistic is just the whole information. So the types of statistics can be divided into descriptive statistics and inferential statistics. Okay, like here, when I describe about the growth, the growth profile, the substrate consumption, and the product formation, of course, all the values here is actually from the mean values. So I do some statistical analysis. When you find the mean value, standard deviation, that is part of statistics, a very simple statistics. So now here I describe the data as it is, right? I describe, okay, the growth is like that. Okay, it reached the highest point over here when it is 12 hour. Okay, and then it starts to decline from maybe 13 hour to 15 hour. Okay, the maximum bioethanol production is, let's say here is 5 gram per liter. So I describe the things as it is. I describe whatever the data that I have. So that kind of statistics, that kind of information is descriptive. Descriptive means that you describe it. Description, it comes from the word describe. Yeah? Describe as it is. So that is this type of data or statistics is called descriptive statistics. So another type is inferential statistics. What is inferential statistics? This is something to do with modeling. Okay, whenever you have the data and you use that data to predict, to give assumption or to give prediction. Okay, maybe like this is not the right data for me for inferential. Maybe in some cases, in some research, you do some sort of modeling and you say that it is predicted that when this happens, this will happen, something like that. Okay, so inferential statistics is something to do with making prediction based on the data that you have. So it's not just about describing whatever that you have. It's different from descriptive statistics, like the case here. Like I describe as it is. I don't talk about the future things. Yeah, but inferential is about making prediction, making assumption about what will happen based on the data that you have. Okay. All right. Not everything. Not the research. Yeah, like this one, it can be descriptive. Perhaps it might not be suitable to be classified as inferential. So the research, it has its own nature lah. Some of them might fall under descriptive statistics. Some of them might fall under inferential statistics. So it doesn't have to be all. It doesn't have to be so and so. So it has its own nature. Okay, right? Like quantitative data and qualitative data lah. So it depends on the research. So some research will generate quantitative and will not generate qualitative. So it's not a fault. Yeah. So yeah, it's something to do with the nature of the research. Okay. Right. I hope that's clear. And then the last step is to process the evidence. Right. So once you have the data, like this one, you have the data. You have seen the trend of the growth, the substrate consumption, and the bioethanol production, right? And you have all these results for all, for example, the range that you have. You have the results for 10%. For this objective, I'm focusing on this example. 10% inoculum density, 15%, 20%. You have that, and you have compared it, right? And same goes for the temperature. For all the temperature that you test, you have the profiles. So from there, you can conclude, you can process the evidence. So that is meant by the last step here, process the evidence, use it to support, disprove your hypothesis, interpret the evidence, and present the findings by communicating with other scientists. Okay, let's go to the first one. You use it to support or disprove your hypothesis. So it means that if, let's say, you do this research and you find out that there is a difference of bioethanol production when you vary the inoculum density from 10 to 20%. Let's say you found out that 15% gave the highest bioethanol production. So it means that now you can relate with your hypothesis. There will be bioproduction using banana fiber, right? That is a general hypothesis, means that you're already accepting the alternative hypothesis. And it's not just saying that there is a production. You found out that the next outcome, whereby you found what is the best inoculum density that give the best production among all the range that you test. Yeah, and the same goes for the temperature. Among all the temperature here, you found out that, let's say, 36 degrees give the best result. So you have that sort of, because based on the data that you have, you compare you process it, and you can make the conclusion. Okay, right. So that is meant by the first point here. You use it to support, disprove your hypothesis. So when you process it, you already know which hypothesis you accept and what are the conclusion, whether or not you answer the objective one, whether or not you answer the objective two. Yeah. So at this step, you also making the conclusion. So when you do the conclusion is always something to be related with the objective. Okay. So based on this one, you compare the data analysis that will bring you to the conclusion. Okay, conclusions. So what needs to be answered or what needs to be stated in your conclusions? You should answer your objective. Answer your objectives, right? So if you have two objectives, you should make sure that in your conclusions, you answer those objectives. Let's say your objective here, you want to investigate the effect of inoculum concentration on bioethanol production using banana fiber, right? You want to study the effect. Okay, so your conclusions is, let's say you found out just now 15% is the best. You can say that there is a relationship. There is an effect when you vary the inoculum density, and in addition to that, you found out that 50% is the best. Inoculum density. Okay? That is your conclusion based on your result. And that is the first objective. And in the second objective, you say that you want to study the effect of temperature. And when you observe, there is a difference, and you find out that, let's say, 36 degrees is the best. So you have to answer it also. You have to say that there is an effect of varying the temperature on the bioethanol production, and the best temperature is 36 degrees. Okay? That answers your objective. If you don't mention those information, it will be hanging. Yeah? If, let's say, I read the report, I expect so what will be achieved from the objectives. Okay. Right. I think that's it. Okay. So process the evidence means that you analyze your data, develop your conclusions, and you relate that with the objectives. Okay. And it's not just that. Okay, once you have it, you also have to disseminate. Yeah, I forgot the last point. Disseminate the final findings. Okay, so once you have completed your research, you do it like you write a report, and how you want to tell people. This is communication, whereas communication comes into play. So you have to write a thesis is also a communication. You have to present your findings or results in the seminar, in the conference, in your thesis, in FYP thesis. When people read later, that is how you communicate to them what you have done during your FYP. Dissertation and thesis is the same. Journal articles, apart from the thesis, you can also write an article. Yeah? Article whereby you submit the article to a journal, and it can reach higher, wider audience. So the difference between thesis and dissertation, some of the universities, they don't publish their thesis, but FYP is not published. Means that it's just for the matter of for you to graduate. It's very hard for others to actually reach that work. So that's why the journal articles is important, because if you write in the journal articles, when you write an article, that article can be accessed by more and more people around the world. So more and more people can get to know your results, get to know your research. Yeah? Books is another way. Yeah? So Viva Virtually, this one is normally for FYP, we don't call it Viva. It's an oral examination for master degree and PhD degree. Yeah, so it's about presentation of the work actually, but they call it Viva, yeah? Whereby they are examiners. For FYP, we don't call it Viva. Yeah. All right, so that's it. That's the 7th step. Process the evidence. Right, so that sum up all the steps in a modern scientific investigation. So I just repeat all the steps. The first one is identify the question, define the problem, find out the things that we want to study, the gap, okay? And from there, you form the hypothesis. From the hypothesis, you predict possible observations or results. Design, and then you design an experiment. You find out what's the variables, how you're going to control the range of the variables. You determine the procedure. Okay, so that's what from 1 to 4 is what you have to write in a research proposal before you enter the lab. And after you have the plan, right, for the FYP, you have to present it to your examiners. And once it has been approved by the examiners, then you carry out the experiment in the lab, right, based on your plan. But it's not based on completely on your plan, but some of the things might be changed. But to say that the proposal is not rigid, some of the things can be changed, and it's fine, as long as it makes sense, lah. So that's why it's called proposal. It's not a definite documentation yet. So you carry out the experiment. So when you carry out the experiment, you have to collect and analyze your data. As I mentioned just now, how you analyze it, it depends on your research. You have to know what kind of analysis, and you have to also know the data, the type of data that you are dealing with, right? So that's why you learn statistics in your first year in order to have understanding about the data analysis, right? And then you process the evidence. You relate with your objective. What is the findings? What is the results that you get? Whether you which hypothesis you accept at the end is based on your work, right? Some of the cases might accept the null hypothesis. Maybe there is no change. So don't be. I mean, there is no hypothesis. There is no bad hypothesis. So if you have to accept the null, that's fine. I mean, that is the fact. Okay, you cannot say that the alternative hypothesis is always the right hypothesis. There is no right and wrong hypothesis. Okay. And then you interpret the evidence and present the findings in the form of reports or presentations. Okay, so that's the communication. That's where the scientific communications comes into play. All right, so that's it for today. I hope that you understand what I have explained here. And the examples that I gave here, I hope that is useful. Any question? Any question, guys?
Doctor,
Yes.
can you repeat again the experimental research for the control scientific research
Control scientific research.
in type of experimental studies?
Great. You mean the... Oh, okay. Control scientific experiment, is it?
Yes, yes.
So it's like how you control the things. So you control variable in order to get the outcomes of your result. So that is basically control scientific experiment. The result depends on your control action, the control action of your work. And how you control it, you vary the variable. Like in the case of here, you vary it from 10%, 15, 20. That is the control. You got it?
Okay. Thank you, doctor.
All right. Okay. So identify controlling as many factors as possible that may affect the outcomes of the study. Factors here is the variable, the things that can influence the result, right? So if you vary like 10 to 50, for example, in this case, the variable, right, from 10 to 20%, there could be a variation in the results, right? So the variation is actually depends on the control action. So that's why it's called controlled scientific experiment. Okay. Any more questions? I have a task for you. If you go to Elite, if you go to Elite, there is a tutorial one. Okay, click on that. Before that, you have selected your group members, right? So please stick with your group for any tasks after this, including the assessment or the one mentioned, the oral presentation, the group assessment, yeah. And this is one of the task tutorial, right? So here I attach an article. You click on this PDF, right? Read the research article, and in your group, you can discuss. And what you have to find out is you have to identify and state what is the research problem stated in the article. What are the hypotheses? Okay, hypotheses are not directly mentioned in the article, but you have to develop based on your understanding of the research. I'm sure that if you understand the research, you can predict. I mean, if let's say you are you can predict the hypothesis. Okay, so you have to develop the hypothesis, both null and alternative hypothesis for the article. You have to find out what is the research questions. Oh, I forgot about the research question. Okay, before that, parameters and their range. What are the parameters to be studied? And the types of data, qualitative or quantitative, or a mixture. Okay, maybe some of the information are qualitative, so you have to mention it. Okay, this bit of information is qualitative data, and these bits of information are quantitative. Okay, I mean, you have to mention it, stack those data. So you have to perhaps extract the information and put it in your tutorial, in your submission. Okay, and this one is to be done in a group. Okay, so research questions, I forgot to explain just now. Okay, what is meant by research question in the case of the examples that in example of let's go back to the example that I showed just now. It's something that you can derive from the hypothesis. Okay. For example, here, okay. So the hypothesis says that there will be the alternative hypothesis is that there will be bioethanol production using banana fiber, right? And you have, let's say in this case, you define the variable inoculum density and also the temperature. So the research question is you question back the things here. Will there be any change of bioethanol production using when you vary? How to say that? Research question. What is the effect of varying the inoculum concentration on the bioethanol production? So that is the research question that can be derived from the first objective. The second objective, what is the effect of the bioethanol production when the temperature is varied between so and so? Okay, that is how the research questions can be developed or can be established. Yeah, I hope that you can relate with the article later. Okay, so basically that's the task is all about. And please submit please use the time maybe, yeah, this week. It's not really much actually because it's you do it in a group, okay, and you just read the articles. And if you understand these things, it's not difficult for you to identify those things. Okay, so identify those things. You just extract the things like data. Okay, you just extract from the article, put it. This is qualitative. This is quantitative. And you have to mention it. Yeah. Parameters. You just identify from the article. Yeah, everything. Right. Please submit by 4th April because we're gonna have the class on the 5th April, right? So I want to discuss it during the class, yeah? So that you know whether your interpretation is correct or not. Right. Okay, that's it for today. Any question? Any question, yes?
So far, nothing, Doctor.
Okay. For the attendance. Check. Okay, I will set the manual attendance for those who did it. Okay, so if there's no more questions, yeah, we can discuss here. All right, okay. I'll set the attendance later for those who need it. Because I have a meeting after this, so I need to do now. All right, so thank you everyone for your attention and for your attendance. Yeah, I'll see you guys next week. Okay, right, don't forget your tutorials. Okay, all right, bye. Have a nice
Thank
day.
you, Doctor.
For today.
Okay,
Thank you, Doctor.
okay, bye.
Thank you, Doctor.
Nggak tahu dia.